Today we will discuss how the immunotherapy efficiency can be improved by targeting immunotherapy along with the ECM stiffness. As we have understood from our previous videos that cancer cells leads to the increase in the ECM stiffness, and that will in return promote the pro-tumorigenic responses. So we will understand the combined effect of this ECM stiffness and targeting ECM stiffness and immunotherapy to improve the efficiency to improve the efficiencies in terms of reducing the tumor cells within the patients. And so what are the problems that occurs when only the immunotherapy is used is that immunotherapy has their checkpoint inhibitors. So checkpoint inhibitors are being blocked uh, by this checkpoint inhibitor that are present on the immune cells are being blocked by the immunotherapy in terms of this anti PD-L1 receptors that will that will interact with the T cells, so that the checkpoint inhibitors will not be able to interact with those checkpoint inhibitors that are PD-L1 that are present on the immune cells. And in that case there is no interactions of the cancer cells with these immune checkpoints with this PD-1 ligands that are present on the immune cells, and the deactivations of the T cells will be suppressed. However, the problem is that in that case is that the stiffness of the around the cancer cells is very high, and because of that the immune cells will not be able to reach to the cancer cells. So this efficiency can be improved by using the agent that can help in reducing the stiffness around the immune cells. So one such idea is that this SNF gene, the lack of SNF genes in the cancer cells will lead to the low production of these PD-L1 receptors. At the same time, the hypoxia condition that are present around the tumor cells, if some hypoxia inhibitors can be used, and those hypoxia inhibitors that further leads to the production of this stiff ECM will be reduced. These hypoxia inhibitors, the HIF1 alpha inhibitors, will along with this PD-L1 uh, antibodies can be used to improve the efficiency of this immune checkpoint blockade therapy, as this HNF alpha inhibitors will reduce the stiffness of the ECM. At the same time, this anti PD-L1 antibodies will prevent the deactivations of the T cells from the cancer cells, and this lack of this SNF alpha will promote or will decrease the amount of this PD-L1 data expressed on the immune cells. So these three things can be targeted to improve this immune checkpoint blockade efficiency. Next one is about the adaptive cell therapies, and this kind of things has observed in this kind of this is the example of the cancer cells where T cells, when they reach to the tumor cells, then it will acquire the pro-tumorigenic properties, and these things, and in order to target this kind of conditions, it's very difficult to directly target the tumor cells. So what they will does is they will target the laminal cells that are present on the periphery of the tumors. For example, these EDA domains and EDB domains. So these things are been targeted along with this is ECM reduced. So in this let me rephrase my sentence. So in the case of this adaptive cell therapy, this EDA and EDB domains are being targeted, and this EDA and EDB domain, what it will does is it will try to reduce the stiffness around the cancer cells. And what has been observed is that by targeting EDA, they can see the reduction in the collagens around the around the cancer cells uh, in this FN cancer disease. And while they are targeting EDP, then they can see the reduction is the reduction in the properties, or reduction in the genes that are related to the metastasis or migrations, such as the epithelial to mesenchymal transitions related genes are being reduced when the EDB or EDB domains are being targeted by these adaptive cell therapies. And when the EDA domains are targeted, the stiffness of this around the tumor cells will be reduced in terms, for example, the collagen ones productions will be reduced. So this targeting therapies can be used so to reduce the ECM stiffness, and thereafter later on this when the cancer-associated fibroblast or CAF envelope is reduced, then the cells then the immune cells that can target the cancer cells can be used, so that the efficiency of those modified CAR T cells will be increased. Instead of directly using those CAR T cells instead of directly using those autologous CAR T cells directly, they can first target this EDA and EDB domain and then use autologous CAR T cells to target the cancer cells. The next one is about this oncolytic virus therapy. So in this case, virus has the cells or the modified cells has the ability to distinguish between cancer cells and the healthy cells and target only the cancer cells. However, in this case also the problem is to reach to the site of the tumors. So these things can be improved. The efficiency of this oncolytic virus therapy can be improved by using the reagents that helps in the ECM degradation, such as hyaluronase reagents that can help in the ECM degradations. Thereby, it will improve both the immune response and also the virus efficiency to detect or to reach to the site of the tumor cells will be improved. So last one is about this therapeutic cancer vaccine. So it is similar to this oncolytic virus more or less. However, in this case the vaccines are used to target the tumors whose amount is very low. It's after, you can say that after chemotherapy, it can be this cancer therapeutic vaccine used so that the tumor regrowth will be reduced. So the similar problems here arises is of the ECM stiffness that will prevent the vaccines to reach to the tumor site. So in this case, to reduce this or to degrade this ECM, the cancer vaccines will contain gene that can produce these hyaluronidase reagents, and that will help in the degradations of this ECM around the tumor cells. At the same time, so that this activated CD8 plus T cells gets activated and can work towards killing the tumor cells. So this is how the immunotherapy's efficiency can be increased if they also take care of this ECM stiffness. So I hope you got just a basic idea how these combined therapies will lead to the increase in their overall impact towards diagnosing the cancers. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.